All right, welcome back. We were just talking about what makes up the DNA backbone. Does anyone remember the six basic parts of DNA before we go on? Yes, please do. All right. Two of the basic parts are right on the board here for you. They are the what and the what. Very close. They are the phosphate group and the deoxyribose. The other parts would be the four bases: cytosine, thymine, adenine, and guanine. We'll get to those soon. Okay, first thing we're doing though is we're looking at the backbone of DNA. We said yesterday that we had a phosphate group and a deoxyribose group come together. They bond, releasing water. Now, the next step is, how do we get two of those to bond? Let me remind you, no notes are necessary at this point. You're not going to be tested over all these minute details. I just want you to understand them so when I explain the bigger picture, you understand where it's coming from. Okay, that sounds good. All right, so here is one of the pieces from the other page. Notice that when it comes in contact with another phosphate deoxyribose group, again, it's going to give off two hydrogens and an oxygen. What is that again? Water, yes. As we see over here on the right side. Now, it does that over and over again. You have thousands of these phosphate deoxyribose groups in a long chain. What part of the DNA is this making up again? If you consider DNA a twisted ladder, the outside, the ridges or whatever, not the legs, not the rungs, it's the sides. All right. So now we've seen how the backbone is made. Now let's talk about how these other four groups, these acids, if you will, start bonding to the backbone. Are you ready? All right. Let's go over adenine and cytosines real quick here. Make sure I'm on my repeat. All right. These nucleotides are what are going to form the rungs of the ladder. Okay? We said there were four: adenine, cytosine, guanine, and thymine. Right now we're going to look at adenine and cytosine. Adenine was one of the double ring nucleotides. See that? Now again, adenine comes in, and what is left off? Water. Beautiful. And now when cytosine comes in, a single ring, it's going to let off water. So then what you're going to have is this nice backbone of the DNA with two half rungs and some water. Now notice where the nucleotides bonded. They both bonded in the same spot. Do you think this means that only specific nucleotides can bond in specific spots? No. No, they can bond in any order. But the order that they bond in is crucial for how the DNA is read. All right, let's move on and see the other side. Before we can go to the other side of the backbone, I want to show you something. One side of the ladder, if you will, goes from phosphate down to the deoxyribose group. The other side goes from the deoxyribose group to the phosphate group. But actually they're the same thing, it's just that it's twisted upside down. Does that make sense? So you have on the left side, let's say you got it going from phosphate group down to deoxyribose. On the other side you have a phosphate on the bottom going up to deoxyribose. Does everyone see that? So now we're looking at the other backbone. that is where we are going to leave off for today. Thank you very much.